Hello and welcome everyone to this podcast. In this podcast, we're going to talk about three broad things. We're going to talk about epigenetic marks, really just introducing it to you in a very general way. And then I want to remind you of the different types of RNA molecules. And then I want to close with some important features or hallmarks of epigenetics. Why is it important to study it? And we've already touched upon some of that earlier in the first two classes, but I want to bring up a couple other things with you. Okay, let's go ahead and get started. So let's talk about these epigenetic marks. And essentially what I'm saying here is what's happening to the genome? How is the genome being labeled in such a way to allow for genes to be turned on or off in such a strong way that it's heritable? So remember, we talked about the genome being the chromatin, that is, in areas of heterochromatin, being wrapped up into these nucleosomes. And that if there are genes found within this heterochromatin, it would not be accessible to enzymes. So enzymes would not be able to access those genes. And when we think about marks, we can think of a few that I'll just introduce to you right now, is that to remove these histones, to mark these histones for removal, we can add acetyl groups to the histone proteins. In doing so, it neutralizes the charge of the histones. So to remind you, DNA is negatively charged, whereas histones are positively charged. And we'll talk more about that later. But by adding an acetyl group to these positive amino acids on the histones, what happens is it neutralizes the histone in the DNA, and it unravels the DNA within the heterochromatin to make the euchromatin. So now I'll just kind of unravel this in a really obnoxious manner here, like so, because the histones have been removed. So that's one kind of mark we can think of. But then really the major mark that I want to focus on for just a little bit here happens at the gene level. So here's a gene, we'll call it your favorite gene, YFG. You can put your favorite gene there, whatever it happens to be. But remember, a part of this gene, it's not something outside of the gene, it's part of the gene, is the promoter. And the promoter, to remind you, is what these enzymes here, by and large, will bind to activate transcription and turn that gene on. What can happen is that the enzymes have access to this promoter because the histones have been removed, but because of methylation patterns on the promoter, it will prevent these enzymes necessary for transcription, and we'll talk about these enzymes next class, and so that will block transcription. What we know is that these epigenetic marks, these methylation patterns, not only do they stop transcription of this gene, but when the cell divides, these methylation patterns persist and will continue to block transcription. So it's heritable. We will spend a lot of time talking about both these concepts, removing and adding these acetyl groups and adding and removing these methyl groups. What I want to do now is spend a little bit of time talking about RNA molecules. If you remember the central dogma, DNA to RNA to protein, a lot of these RNAs are involved in this process, particularly the ones you are probably the most familiar with. And we know that this process is going from DNA to RNA. What we're really talking about here with the RNA is messenger RNA. So this is our messenger RNA. transfer RNA, all the other RNA molecules are also transcribed. But when we think about what this initial central dogma stated, it was going from DNA to RNA to protein. In this case, we're talking about messenger RNAs. But I do also want to stress again that when we make all the other RNA molecules we're going to talk about, that is still transcription and it's still coded by the DNA. Then in the process of making the protein, two of our other RNA partners come into play. Transfer RNA. And remember, this is the molecule that brings in the amino acid during translation because it matches up along the messenger RNA, like so. Let me make at this region here between the anticodon and the codon. And this all happens within the confines of the ribosome. And to remind you, a large part of the ribosome is made up of ribosomal RNA, or what you might remember as simply rRNA. Now, I want to bring up some other RNA molecules that you may have been taught about in some other classes, or maybe you haven't been. So I want to make sure these are all on our radar as well, because they all play a role in gene expression. The next one I want to talk about are these small nuclear RNA, or SNRNA. Remember that when DNA is transcribed into messenger RNA, what is produced is an immature RNA that contains a mixture of exons and introns. These introns have to be removed to produce a mature messenger RNA that contains just these three exons. It is these three exons that are important in making the protein. The introns do not code for any part of that protein. And these small nuclear RNAs, they play a role in this processing of this immature RNA into the mature RNA. The next one I want to talk about are microRNA. Sometimes we call these interfering RNA. In essence, what these microRNAs do is they inhibit the translation of messenger RNA, and only messenger RNA. And to be very brief about it, we could spend lectures talking about this, but I just want to kind of give you a flavor of what they do. Is if you have a messenger RNA here with its G cap and its poly A tail on them, so it's a mature messenger RNA. So what these microRNAs do is they will come along here, they form this duplex structure here. They will bind to specific messenger RNA molecules. They will recruit enzymes. We'll talk more about that later. And as they recruit these enzymes will degrade the messenger RNA. So it results in a degraded messenger RNA. This is a very important process of inhibiting the ultimate expression of a gene. You don't affect the DNA itself, but the messenger RNA, you destroy that, and now you no longer have the ultimate expression of that gene. I also asked you to know what a small interfering RNA is. So let's talk about that real quick. Which is often called siRNA. And it does the same thing that a microRNA is in general. They have a different kind of specificity, but they still degrade messenger RNAs. The big difference is that the small interfering RNAs typically come from outside the cell. Meaning the microRNA, these are made by the cell itself to control gene expression, whereas the small interfering RNA, the siRNA, are introduced from outside the cell. Many viruses will use this approach to degrade certain gene expression of their host cell. That's the big difference that you should focus on. The next one I want to talk about, next RNA I want to talk about, are these ones called piRNA, or sometimes we call them piRNA. And these are really important in epigenetics, maybe more so than the other ones that we've talked about, because they seem to play a role in gene expression in the germline, particularly during spermatogenesis, or at least there's more evidence there. But their goal is similar to the microRNA in that they have a purpose in degrading specific RNA targets. The last one I want to talk about is a very important one also in epigenetics, and these are called long non-coding RNA. And we'll talk more about this throughout the semester, but I want to provide an example of what they can do. As the name implies, they don't code for anything, but they're as long as you might suspect a messenger RNA to be, and in some cases a lot longer than that. And let's talk about X inactivation, just briefly. We'll come back to it later. We know that in females there are two X chromosomes. A female with two X chromosomes, and these are those two X chromosomes. We know that through the process of X inactivation, one of these X chromosomes gets inactivated, and it gets inactivated by these non-coding RNAs, and it forms what I try to show here as a bar body. What happens then is there's this gene here, what causes this I should say, there's this gene on both X chromosomes, but only one of them is activated, and it's called the XIST gene. And it produces a messenger RNA. I'm sorry. It produces these long non-coding RNAs. And what it will do is it's being produced, it will wrap itself around one of these X chromosomes and compress it like so. And in doing so, it downregulates, turns off gene expression of the X chromosome. Now, it doesn't completely turn every gene off. There are some genes that stay active, but by and large, it turns most of those genes off. All right, I'd like to close this podcast talking about some important consequences of epigenetics. And I want to talk a little bit about evolution. And if you think about Darwin and Mendel's findings, what it would suggest is that the phenotype of animals and offspring are largely controlled by the genes, and largely the sequence of those genes. There was this other guy, his name was Lamarck. And Lamarck thought that, we've talked about this term before, but the inheritance acquired traits, meaning that traits that an organism acquires throughout their lifetime because of experiences from the environment, they could pass those on to the next generation independent of their genes. And over many, many years, Lamarck and his ideas were largely discredited. And for the most part to this day, we don't really think so much of this inheritance of acquired traits as being a possibility. However, as we start to explore epigenetics, we're starting to see how the environment, what an organism experiences, can affect what's inherited to their offspring, or the phenotype that their offspring will have, independent of the genes. Now, I think these findings that were discovered with epigenetics certainly don't disprove Darwin or Mendel. Rather, they enhance their findings. And it brings a little bit more credibility to some of Lamarck's ideas. Not all of them, but maybe some of them. And some of these observations, your book highlights a couple. I'm just going to mention them, but I'll encourage you to kind of read up on them a little bit more if you're interested in finding out more about them. And one of these was the Dutch Hunger Winter of 1944. It resulted from a blockade that the Nazis set up. So the Nazis had a blockade against Holland. And it prevented a lot of food from entering into this country. In addition, at this time, unfortunately, there was also a very harsh winter. And as a result of both of these things here, there was widespread famine. The children born after this period, so I'll say offspring, even when they were born after the blockade and once food had returned to the area, they had decreased lifespans and more susceptible to diseases like diabetes, heart disease, and your book mentions some others like schizophrenia and obesity. Now, what was even more interesting from the epigenetics point of view is that the grandchildren, so their parents didn't experience this famine or this blockade, but their grandparents did, also had these same increases. Somehow the grandmother's diet affected the next generation and the subsequent generation. If everything was about the genes, this would not explain it. There has to be something else that goes above the genetics of it. What is that? Well, that's where we will bring in epigenetics. Those genes are somehow being modified, but not at the level of the sequence. The other example that your book brings about is breast cancer. We know that a very small percentage of people with breast cancer have an inheritance pattern, meaning that about 10% of all cases are linked to inheritance, meaning about 10% of the cases they will have a mother or a father who have also had breast cancer, or maybe a grandmother or an aunt that had breast cancer. The other 90%, as far as we know, no inheritance factor. So there have been a lot of studies on this. Your book briefly highlights a couple of them. One where they give mice or rats a high-fat diet, and this led to female offspring to have an increase in breast cancer. However, in this particular study, the next generation did not have that same effect. However, another experiment using another cancer-causing agent called ethinyl estradiol had the same effect, female offspring to have an increase in breast cancer, but it was also seen in granddaughters. Again, it didn't change the sequence of the genes or the DNA, it just changed the ultimate phenotype. How some of this happens, we don't know. And we'll talk more about this throughout the semester. There are some very interesting studies done where the epigenome seems to be more important in causing cancer than the actual gene mutations. They're both important, they both play a role, but there are some studies that suggest the epigenome has a more drastic effect in causing cancer. Okay, let's end here with a quick summary. In this podcast, we talked about epigenetic marks, and we focused primarily on acetylation with acetyl groups and methylation with methyl groups. Acetyl was on the protein histone, methyl was actually on the DNA. We then talked about all the kinds of RNA. We reviewed the ones that we were most familiar with, the messenger RNA, the ribosomal RNA, and the tRNA, but I introduced some of these small interfering RNA molecules, particularly microRNA and the Pee-wee RNA. And the latter, the Pee-wee RNA, seems to have a role in epigenetics. And then we talked about the large non-coding RNAs and how they're responsible for gene expression, and we gave the example of X inactivation. And then we just closed with a discussion of the importance of epigenetics. How it's not all about the genes when it comes to inheritance, that there is some strong evidence suggesting that it's beyond just the gene. There is certainly an inheritance of genes, and mutations do get passed on, but there's something above that that we also need to pay attention to, and that is the focus of this class. All right, if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.